Our ability to hear depends on sensing minute movements caused by sound vibrations. When sound waves enter our ears, they journey to a spiral-shaped structure called the cochlea, where they make special cells vibrate. This system is astonishingly sensitive. The faintest whispers we hear make these cells move by just nanometers. This is the scale of individual molecules. The DNA you see here is about 2 nanometers wide. Compare this to a single strand of your hair, about 30,000 times wider. In this video, we'll dive into the fascinating inner workings of our ears. We'll discover how these cells detect the subtlest of vibrations and convert them into electrical signals that our brain can interpret. To focus on essential concepts, some technical names and details will be simplified or omitted. We'll see how our brains process these signals, what occurs during hearing loss, and even how our ears can create sounds. The cochlea, located in our inner ear, contains auditory hair cells. These are named for the hair-like structures called stereocilia at their tops. Sound causes these stereocilia to sway, a movement we're visualizing slowed by several hundred times. This movement triggers electrical signals within the cells represented by changing colors, redder being more positive and blue more negative. The quieter the sound, the less they move and the weaker the electric signal produced. Even at the current magnification, the softest sounds we can hear produce movement of less than a pixel. We zoom in further to see how we achieve such sensitivity in hearing. Strung between the stereocilia are thin filaments called tip links that are part of a mechanical system essential for our ability to hear. Each tip link is connected to a protein called an ion channel. When sound moves the stereocilia, the tip link pulls open the gate at the top of the tunnel-shaped ion channel. When open, positive charges rush into the cell, producing an electric signal. These positive charges are ions like potassium and calcium, shown as red dots. Movement in the other direction relieves the tip link's tension, closing the ion channels. The positive charges are removed from the cell elsewhere, returning it to its initial state. This elegant system translates sound into an electric signal by relying on tiny switches. To perceive sound, the electrical signal must be relayed to the brain. There are 2 types of hair cells, outer and inner. Only the inner hair cells pass on their information to the brain. Outer hair cells play a different role, which we'll return to later. Inner hair cells relay their electric signals to adjacent cells called spiral ganglion neurons. When the inner hair cells become more positive, they trigger nearby spiral neurons to produce discrete electrical pulses called action potentials. These action potentials rapidly travel along the cell, carrying the information to the brain. Every sound you perceive is encoded into sequences of action potentials, which our brain interprets to give us the experience of hearing. The specific pattern of these action potentials somehow encodes everything we hear. But how can a digital sequence of action potentials relay the full richness of sound? Complex sounds like voice or music have variations in frequency, volume, and timing. Let's see how each of these is represented by action potentials, starting with frequency. We can hear sounds from 20 Hz to 20,000 Hz, though this range decreases as we age. With approximately 1,500 inner hair cells per ear, each is tuned to a narrow slice of this spectrum. They are arranged like strings in a piano. Cells sensitive to high notes or frequencies sit at one end of the cochlea. Those attuned to the lower notes are at the other end. Play a brief, high-pitched sound in a quiet room. This stimulates some hair cells that trigger action potentials in spiral neurons. These action potentials race down the neurons, traveling to the brain. Now play a lower pitch. Different cells generate action potentials. The frequency of the sound, then, is indicated by which neurons generate action potentials. Every frequency is assigned to specific cells. What if the sound is louder? In response, the hair cells generate larger responses, resulting in spiral neurons firing more action potentials. The volume of a sound is indicated by the rate of action potentials. Finally, let's consider timing. For sharp, sudden sounds, the action potentials start when the sound first arrives. Thus, the timing of the action potentials reflects the timing of the sound. Real-world sounds are more than single tones, and the encoding of sounds into action potentials is more complex. But the key thing is that all information is represented through trains of action potentials, their specific rate, timing, and origin carry the rich details of sound. In addition to the nanometer scale sensitivity of hair cells, their time precision is equally astonishing. Consider watching a video at 30 frames per second. We don't notice the 33 millisecond gap between each frame. Yet in ideal conditions, we can sense differences in sound timing in microseconds. Imagine a rustling noise nearby, and you try to determine its direction. How do you do that? You use several cues, but we'll focus on the time difference between sound reaching your left and right ears. For a sound coming from directly in front, both ears hear it simultaneously. However, if it's coming from the right ear hears it a few hundred microseconds before the left. Each direction has a different delay, with research revealing that some people can detect time differences of 10 microseconds or less to localize sound to within a few degrees. Let's explore a neural circuit that measures this time difference. Action potentials from our ears travel through several pathways in our brain. One destination is the structure shown here. The bottom cell carries the first action potential triggered by sounds from the right ear, while the top carries that from the left. Action potentials travel along these cells at several meters per second. Their information feeds into a middle row of cells that function as coincidence detectors. These cells fire action potentials when they receive simultaneous input from both ears. For a sound coming from straight ahead, both ears hear the sound at the same moment, prompting action potentials in the top and bottom cells. They meet at a particular coincidence detector, which then fires its action potential. But shift the sound slightly to the right and things change. The right ear responds first, followed by the left after a brief delay. Now the action potentials meet at a different coincidence detector. This signals a sound coming from further to the right. These coincidence detectors pass on the sound's timing information to other parts of our brain. The brain can determine the direction of the sound from which cells are firing. This doesn't give us a full 3D direction, like whether the sound is above, below, in front, or behind. For that, our brain uses other cues, such as subtle frequency changes. Disruptions or alterations in the action potentials carried within the brain causes hearing problems such as tinnitus, characterized by the perception of noise or ringing in the ears. This can occur when spiral neurons fire action potentials when they shouldn't. Spontaneous firing of action potentials can lead to phantom auditory sensations because our brains are wired to interpret action potentials from the spiral neurons as sound. However, tinnitus can stem from several other causes. Almost everyone experiences age-related hearing loss. Sustained exposure to loud noises can kill hair cells, particularly those sensitive to high frequencies. We can't regrow dead hair cells, so their loss is permanent and cumulative. Suppose we lose our inner hair cells that detect high frequencies. In their absence, spiral neurons don't generate action potentials. Consequently, the brain never perceives high-frequency sound, and we are deaf to these sounds. Most age-related hearing loss stems from damage to the outer rather than the inner hair cells. Inner hair cells function as microphones. They convert sound into an electrical signal. Outer hair cells don't trigger action potentials in spiral neurons to convey information to the brain. They're amplifiers, are built-in hearing aids that boost quiet sounds. To understand their importance, let's watch two responses to the same sound, one with functioning outer hair cells on the left and one without on the right. Notice the larger amplitude motion with working outer hair cells. The difference may seem minor, but it's significant on the nanometer scale detected by inner hair cells. Without outer hair cells, the quietest discernible sound shifts from a whisper to having to shout loud enough for Grandpa to hear you. How do outer hair cells amplify sound? Initially, outer hair cells work just like the inner. Sound moves the stereocilia. This induces tension in the tip links that opens ion channels, making the cell more positive. When the sound moves the stereocilia in the other direction, channels close and the cell becomes more negative. This is where outer hair cells are different. When positively charged, they shorten, pulling on the membrane above and boosting the movement. When they become negative, they extend, again boosting the movement. In both cases, the sound movement is amplified. This mechanism has been experimentally verified. We can place an electrode in an outer hair cell and connect it to an electronic signal generator. When we apply a changing voltage, the outer hair cell visibly alters its length in response. These cells respond at incredible speeds, able to move at the upper limit of our hearing at 20 kHz. The mechanism of this movement is not fully known, but a protein called prestin plays a vital role. Prestin is the opposite of the ion channels in the stereocilia. It's a molecular motor that converts electrical signals into movement. When a cell is positive, prestin is in a compact shape. When the cell becomes more negative, prestin expands and makes the cell taller. When the cell is more positive, prestin contracts. The motion of a single prestin is tiny, but the membrane is densely packed with prestin. They all sense the same electric signal and synchronize their expansion and contraction, working in unison to make the cell taller and shorter. The mechanical motion of outer hair cells can produce sound in some conditions. If the outer hair cells move when they shouldn't, they can produce motion in the cochlea that produces pressure waves. These travel out of the ear to produce faint sound. Rarely, tinnitus may be caused by inappropriate activation of outer hair cells where the ear emits noise, and the ability of the ear to emit sound is clinically useful. If you turn up your computer speakers too loud, you can hear distortions or alterations in the sound. Outer hair cells also produce distortions. In a hearing test called autoacoustic emission testing, specific tone pairs are played into the ear, and a sensitive microphone listens for these distortions. Imperfect outer hair cell amplification causes the ear to emit faint sounds of different frequencies than those played into the ear. This is a functional test of outer hair cells. When they aren't working, these distortions are absent and no sound is emitted. We've seen how cell processes are mechanical processes, and how we can apply our physical intuition to understand biology. I hope this can be a starting point for further questions and exploration. How are electric signals processed and passed between cells in the brain? What happens next? Can hearing loss be fixed? And there's more we didn't cover, details rich with greater complexity and elegance. At the molecular level, everything is in constant random motion. This is not from sound, but heat. At body temperature, cell membranes are fluid, with molecules in a whirlwind of collision. I want to end with some puzzles. There is significant random motion even in complete silence. I didn't show this earlier, but it is present in hair cells. How might this thermal motion affect hearing? And notice this connection here. This anchors the ion channel to the cytoskeleton, a semi-rigid scaffolding that helps the cell maintain its shape. Is this connection needed, and what does it do? Can you think of ways to reduce the effect of random noise? There's a hint for one mechanism that you can see right now. If you're interested in learning more, check out my previous videos on vision. You'll see the astonishing sensitivity of our eyes and why there's a faint shimmering when we look around in the dark. And vision, like hearing, starts with a tiny mechanical change. Our brain and senses rely on noticing not just single cells, but individual molecules.